Welcome back to General Embryology series. This lecture is designed to discuss meiosis in brief and its relation to gametogenesis. So, we will not go in depth in explanation of meiosis. But before starting, I need to discuss two important points. The first point is general structure of chromosome and chromosome backing. Second is the major differences between mitosis and meiosis. So now about the general structure of chromosome and chromosomal backing. Here in this figure, we should differentiate between single-stranded chromosome and double-stranded chromosome. Single-stranded chromosome, which is formed of one chromatid. This is described as S chromosome, while chromosome with two chromatids attached with each other to the centromere, this is known as D chromosome. D chromosome normally seen after replication of the cell. So here, this is to describe the chromosome and the chromatin. So the chromosome, as the cell prepares to undergo mitosis or meiosis, the chromatin fibers become extremely condensed, forming the chromosome, reaching maximum condensation during metaphase. The human genome is made up of 46 chromosomes, 23 homologous pairs of chromosomes, one pair from the father and another pair from the mother. These 23 homologous pairs are classified into autosomes or somatic chromosomes, which are 22 pairs, and single pair of 6 chromosomes. The single pair of the 6 chromosomes is the pair that determines the sex of the fetus. So if the fetus is female, so the sex hormone, the sex chromosome, is represented by 2 X chromosome XX. Whereas the single pair of male sex chromosome is represented by an X chromosome and Y chromosome. So it will be XY. So now what about the structure and the backing? How I can back the chromosome into the nucleus? So several orders of DNA backing occur in chromatin and during chromatin condensation of mitotic prophase. The top, this drawing or this image showing 2 nanometer DNA double helix. So this is the DNA double helix. It is 2 nanometer DNA double helix, followed by association of DNA with histones to form 11 nanometer filaments of nucleosomes connected by DNA. So here each each molecule, such molecule is known as nucleosome. So the nucleosome is the DNA associated with histone. Then the nucleosomes on the DNA, then the nucleosomes in the DNA interact in a manner not well understood till now to form a more compact 30 nanometer fibers, which is this stage. For the transcription, DNA forms loops that remain tethered to and stabilized by interactions with protein scaffold that eventually make up a central framework at the long axis of each chromosome, which is this stage. Heterochromatin is not transcribed and remains more highly condensed. So here in this figure, we have two types of chromatin. This is the heterochromatin, and this one is the euchromatin. Heterochromatin is the inactive part of the chromatin which is not undergo transcription, while the euchromatin, which is the thin form of the chromatin, is the part which is undergo the transcription. Then finally, here in this is in the bottom of this figure, this shows a metaphase chromosome with maximum backing of the DNA. So now the metaphase chromosome consists of two chromatids held together at a constriction. This constriction is known as centromere. Okay. Now about the major differences between as a summary, meiosis and mitosis share many aspects of chromatin condensation and separation. But there is major differences between meiosis and mitosis. Mitosis is one cell division that produces two diploid cells, while meiosis, as we will mention later inshallah, involves two cell divisions and produces four haploid cells. During meiosis, there will be crossing over. I mean by the crossing over is the new combinations of genes are produced, and every haploid cell is genetically unique. So mitosis yields two cells which are of the same genetic composition, while meiosis leads to four cells which are genetically different. And meiosis. One of the meiosis is divided into two meiotic divisions. First meiotic division and second meiotic division. So what are the events, or what is the aim from the first meiotic division? The first meiotic division, the aim is to reduce the number of the chromosomes to the half, from 46 D chromosomes to 23 D chromosomes. Reduction of the amount from the double number to the normal amount of the chromosome. Double amount, this is what I will explain in the next slide. As any cell before entering cell division, the number of the chromosomes should be duplicated. So here in the first meiotic division, it is characterized by duplication of the amount of the DNA. So now each cell contains the diploid number or double the number of the amount of the DNA, and the pairing of the homologous chromosomes. The 46D chromosome in each cell arrange themselves into 23 pairs. The pairing is point to point except for X and Y chromosome. And the first meiotic division is characterized by exchange of genes, which is crossing over. I mean interchange of chromatid segments between the two homologous chromosomes. And by the end of the first meiotic division, there will be separation of the chromosomes. The members of each pair separate into a new cell. So, resulting is two daughter cells. Each cell contains 23 D chromosomes, I mean double-stranded, with normal amount of DNA. So this is why here this slide represent how the chromosomes are first single-stranded, then with replication it will be double-stranded, and here this is the stage of crossing over. So now these chromosomes acquire new genes construction by exchanging of these segments between homologous chromosomes. Then by the end of the cell division, we will have new cells, these cells with new genetically structure. Here I need to differentiate between 46 D chromosomes, 23 D chromosomes, and 23 S chromosomes. D indicates double-stranded chromosome, while S indicates single-stranded chromosome. We should put in mind that at the beginning of any cell division, and due to the interphase which proceed or which prior to any cell division, the chromosomes should be duplicated. So it will be, instead of 46 chromosome, it will 46 single-stranded chromosome, it will be 46 double-stranded chromosome. So 46 double-stranded chromosome, this is at the beginning of any cell division. So at the end of the first meiotic division, there will be a reduction in the number of the chromosome. So instead of 36 D chromosome, it will be 23 D chromosome. This is by the end of mitosis and by the end of the first meiotic division. If the cell is germ cell and will enter the second meiotic division, there will be decrease in the volume of the chromosome, in which it will be converted from 23 D chromosome to be 23 S chromosome. I mean here it will be 23 single-stranded chromosome. This note is very important to understand the process of meiosis. Then what about the second meiotic division? The second meiotic division, the aim of the second meiotic division, as we mentioned in the previous slide, is to convert the 23D chromosome to 23S chromosome. This is the aim. Why? Because of the when one germ cell with 23S chromosome is fertilized with 23S chromosome, now we will restore again the 46S chromosomes which present in any cell. Then before the cell, these 46S chromosomes will be converted into 46D chromosome to start the new cell division cycle. And so in this phase of second meiotic division, there is no DNA duplication, there is no crossing over occur, and now the 23D chromosome, which result from the first meiotic division, will be divided through the centromere into 23S chromosome or single-stranded chromosome. So the result of meiotic division will lead to the formation of four daughter cells. Each cell contains 23S chromosome. In male, the daughter cells are known as primary spermatocytes. So the primary spermatocytes will be 4 primary spermatocytes, but 2 primary spermatocytes will be 22 plus X, and the 2 primary spermatocytes will be 22 plus Y chromosome. While in female, the net result are 4 primary oocytes, and these primary oocytes all contains the same number of chromosome, 22 plus X chromosome, which this means that the 4 daughter cells in female are typical. Again, I repeated this slide again because of the this slide is considered as the cornerstone to understand the cell division. Thank you for your attention. Thank you for your attendance. See you in next video. Good luck and goodbye.